Hi, I'm Lucy Brown, and I'm here with Dr. Martin Smith in the Earth Sciences Department to talk to him about his most recent publication in Nature. Hi, would you like to give us a basic summary of your paper?
Sure, so we've been looking at a group of really weird animals that were alive before the dinosaurs, so sort of 200 to 500 million years ago. And these are animals that have been really confusing for 175 years since they were first discovered. And we've finally been able to work out what exactly these organisms are.
So what are these organisms?
So these organisms are a group called hyoliths, and they are essentially sort of small organisms, a couple of centimetres long, that would have lived in a shell that looks a bit like an ice cream cone, and the shell would have had a little lid on it, like a dustbin lid, and out of the side of the little gap would have been two little stilt-like things, a little bit like tusks. So really weird organisms that don't quite look like anything that's alive today.
What do they use these tusks for? How do they operate?
Well, this was one of the big questions. There were sort of lots of things about these organisms we didn't know. So normally when you're looking at fossils, you think, okay, what is this? And you can compare it to something that's alive today. And you think, okay, a dinosaur is a little bit like a crocodile or a little bit like a bird. And we can use those modern analogues to interpret the organisms. With hyoliths, there's nothing alive today that we could say, okay, it looks like this, so we can work out what it looked like or what it did. So we were stuck, really, for 175 years. All we had were the shells, the four little pieces that stuck together somehow, and we didn't know anything about the animal that was living inside those shells.
How has your recent discovery made a difference to the last 175 years?
So what we've discovered is a set of hyolith fossils that, instead of just being the shells, actually have the body itself preserved. So you can see tentacles, you can see muscles, you can see internal organs, and you can finally put together the anatomy of these organisms.
So how has that changed how you viewed these organisms previously?
So for the first time we're able to say what these organisms were, so we can reconstruct, most importantly, their feeding mechanism. So they had, out of this, the two shells would have come a tentaculate feeding apparatus, so a suite of 12 to 16 tentacles that would have surrounded the mouth, and those tentacles would have been used for grabbing food out of the water, in essence, to feed. So they would have sat still on the seafloor and grabbed food. And why that's important is that there aren't many groups today that have got an exactly equivalent feeding setup. And in fact, we can tell that this is what we call a lophophore, which is a scientific term for an organism made of hollow tentacles that surround the mouth. And a lophophore is only found in a couple of groups of organisms today.
Is that why you thought they were in the mollusk group, because of the...
So we didn't used to really know what hyoliths were, so there were a couple of possibilities. One possibility was that they were mollusks, so things like snails and squid and various other organisms. And the reason for thinking they might be mollusks is essentially that they had a shell, and certain properties of the way that shell was made up looked a little bit like the shells in mollusks, but not really anything like anything else. An alternative idea is actually, because the shell doesn't really look that much like a mollusk shell, maybe hyoliths were a completely separate lineage on the tree of life, so a failed evolutionary experiment that's very distantly related to anything alive today.
Just for everybody watching, what makes a mollusk? What is a mollusk?
So a mollusk, there's not a single character that makes a mollusk a mollusk. They're a very diverse group, and there's all sorts of different body plans within it, sort of ranging from snails to slugs to squid to tusk shells, and there's a huge diversity there. Most of them have a feeding apparatus called a radula, which is like a conveyor belt of teeth that they use to rasp at food on the seafloor or on the land. Most of them have shells, or there are groups that don't have shells. Most of them have a creeping foot that they use for getting around, although there are some exceptions to that. But they're one of the the second most diverse subgroup of the animal kingdom.
And so you said they could be in a separate group altogether, but you now think that they're in the brachiopod group.
That's right. So the brachiopods are a group that are perhaps unfamiliar to in everyday life, but they're quite diverse in the deep sea today. And the Romans actually knew a lot about brachiopods. They look a lot like a Roman oil lamp, and so they were called the lamp shells by the Romans. And they're very familiar to paleontologists. They're one of the most common fossil groups in the fossil record.
Is there anything today that we would recognise as a brachiopod, or looks like a brachiopod?
Well, so brachiopods that are alive today are, they look a little bit like clams, they look a bit like shellfish, and they're considered a delicacy in various parts of the Far East, I think. But in Western society they're less familiar, perhaps. There's another group called the horseshoe worms that you can find in rock pools around the British coast that have got little stalks and then a little lophophore, again, this tentacular feeding crown, but no shells.
And does this change how we see brachiopods and the evolution of brachiopods?
Yeah, so there's a really big problem in paleontology, which is where the modern animal body plans came from. And if you look at the fossil record, it almost looks like brachiopods, mollusks, and lots of other groups appear overnight. There's no clear precursors. There's a very rapid change in the fossil record. And it did look, when we thought hyoliths might be a separate group, that actually the hyoliths were another group appearing out of nowhere. But what we can start to do now is we can link hyoliths to brachiopods, and hyoliths can tell us something about the origin of the brachiopod body plan. So instead of overnight suddenly brachiopods appearing, we see first of all the hyoliths appear in the fossil record, and then we can start to see or speculate on some mechanisms that might change from a hyolith, how a hyolith might evolve into a brachiopod. And so we're breaking down that sudden appearance into a series of more gradual evolutionary steps.
And so maybe you said this, but so there's no definition for a hyolith, but it's more what it looks like.
Yeah, so in paleontology, because we only have fossils, we have to essentially define groups on their morphology. And hyoliths have this distinct morphology of a cone, a lid, and these two tusks. So that together is what makes a hyolith a hyolith. But rather than thinking of the hyoliths as a separate group that only includes hyoliths, perhaps we can consider hyoliths actually to be a sort of a grade and a broad evolutionary spectrum, and some hyoliths went on to go extinct. Other hyoliths perhaps were ancestors to the brachiopods that are alive today.
Could you tell us anything about the fossils that we have here today that relates to hyoliths?
Yeah, so we've got a couple of fossils. So we've got a fossil, this is a mollusk fossil, this is actually an ammonite, and you can see it's got a coiled shell that curves round, and this would originally have been a squid-like organism that would have had sort of a set of tentacles coming out here. It would have used for feeding and had jet propulsion. Hyoliths have been linked to the evolution of this group, the cephalopods, that includes the squid and the ammonites, on the basis of similarities between their shells. But we can now show actually their morphology, the morphology of the bodies themselves is very different, and so we can rule out that link between mollusks such as the ammonites and hyoliths.
So but the hyoliths still have...
So the hyolith has tentacles, but what we can tell now we have the soft anatomy is that the tentacles have a very different origin. So developmentally, the tentacles would arise around the mouth, whereas in a cephalopod, in a squid, like this one, yes. The tentacles would evolve, actually have an origin within the foot of the animal. So it's a mollusk, it's got a foot, and little bits of the foot grow out to become tentacles. So they look the same, but actually it's very independent evolutionary origins. Okay. The other fossil that we've got is, this is a brachiopod, this is a very nice sporiphorid, and what you can see about it, it's got two shells, it's got a top shell and it's got a bottom shell, and within these shells there would have been a cone-shaped structure that would have supported lots of little tentacles, would have supported the lophophore in essence. And so this brachiopod, we could see the water would come in and go through the lophophore and be evacuated from the shell. And it looks quite different from a hyolith, which was just a simple cone, but because it's got that lophophore inside it, we can see an evolutionary similarity.
So it's the soft structure inside that is the same between them?
That's it. So they both have a tentaculate feeding apparatus that has little tentacles that have the same evolutionary origin and move food towards the mouth, have the same role.
So this seems to be a pretty complicated paper with pretty complex terminology. I'm just going to read a sentence quoted from the paper in Nature. We reconstruct Haplophrenitis as a semi-sessile, epibenthic suspension feeder that could use its helens to elevate its tubular body above the sea floor. Would you be able to tell us what that means in English?
Yeah, so we've condensed an awful lot of ideas into a very little bit of sort of technical information. I think maybe a picture speaks 1,000 words in English. So we've got a reconstruction here of the fossil Haplophrenitis. And what we can see, we've got the shell here, we've got the little lid on the top, and these tusks along the side, which are called helens, poetically enough. The sort of tusk-like thing, that's right. So what Haplophrenitis would have done, it would have been stationary. It has been thought these helens would have been used like oars to row itself around the sea floor, one of the more sort of far-out ideas. But we know that's not the case now from the way they fit together into the skeleton. It seems these were used as stilts to elevate Haplophrenitis, this particular species of hyolith that we studied, above the sea floor. And we can work out now why it might have wanted to do that. And that again comes back to this lophophore, the feeding apparatus. If you're trying to pick out little bits of organic matter from the seawater as currents are maybe drifting it by, you don't want to be down right on the mud where you're going to be surrounded by lots of little muddy particles that aren't particularly tasty. So it could be that it used these helens to hold itself up a little bit, and then it could get that clean water with little bits of plankton or microorganisms that it could then pluck out without clogging its feeding apparatus up with mud.
I see. And that's what that sentence meant.
Yeah, that's right, in essence. So we can reconstruct how hyoliths fed, essentially. We didn't know whether they were predators, scavengers, carnivores, but now we can see this feeding apparatus. We can see they stayed still, sat in place and waited for the food to come to them.
I see. So for someone without a science background, why does this matter? Why is this important in this whole scheme of things?
So there are various sort of questions that this helps us to answer. I mean, for me, it's just a real mystery. We've got these fossils that are really common for 250 million years, and we've no idea what they are. And it sort of seems we ought to have some basic understanding of such a rich part of life's ancient diversity. There are other things that are interesting as well when it comes to looking at how the diversity of life came to be. I think this is a question that a lot of people are very interested in. And we really don't understand, to some extent, this rapid burst. So it used to look like there were lots and lots of different organisms appearing out of nowhere. And some people said, well, maybe actually this type of evolution we see today can't explain that. How do we explain that? What other evolutionary mechanisms could be involved? Putting hyoliths in their place in the evolutionary tree helps to constrain how body plans were built up out of the much simpler ancestors that came before. And the other thing that's interesting about hyoliths is because they were such an important part of the ancient oceans, they were playing an important ecological role. Hyoliths go extinct in the biggest mass extinction of all times, 250 million years ago, and whatever the hyoliths were doing is gone. And if we didn't, now we know what hyoliths were doing, we can say, okay, how long did it take for that role in the ecosystem to be filled again once life recovered from this mass extinction? And well, we're undergoing a mass extinction event at the moment. And so it's really important to understand past extinction events so we can see how big they were, how life recovered and how life adapted, in order to understand what we might want to do to mitigate the catastrophe of extinctions that are occurring today.
It's been really fascinating, and thank you. Thank you.
You're welcome.